In this video, we'll talk about the microbial culture media. We'll focus on the frequently used culture media in medical laboratories. Microbes need nutrients to grow, just like any other organism. And we'll classify the microbial culture media on basis of different criteria. We can classify them on basis of their chemical composition. There would be different classes like simple media, defined media, or even complex media. There could be classification which is based on the physical nature of the media, like whether it is solid or liquid. There could be classification on basis of their function, such as supportive media, enriched, selective, or differential media. In this video, we'll talk about some popular media that are frequently used and which are of interest for medical pathologists. So first we'll talk about simple or basal media. Here, these kind of media are used routinely in labs and they are generally used to grow non-fastidious bacteria like Escherichia coli, Enterobacter, and Pseudomonas aeruginosa. So they have very simple composition. They need to have a carbon source, a nitrogen source, and some salts. So generally they have beef extract or yeast extract, which can serve as nitrogen source, peptone, and also they have salt such as sodium chloride. Sometimes they have solidifying agent such as agar if we are talking about a solid media. So these kind of media is used to primarily isolate microbes from patient sample or let's say from other sources. Example of these media includes nutrient broth, nutrient agar, peptone water, etc. Now the defined media or synthetic media are slightly different from the simple media. Both these media need to have carbon and nitrogen sources. But the difference between these two media is in defined media, all these sources are really defined. For example, in simple media, beef extract or yeast extract are kind of like undefined source of nitrogen. But in case of defined media, all these proportions has to be exact. Let's talk about enriched media. As the name suggests, there are additional nutrients which are added in this media, such as blood, serum, egg yolk, etc. So enriched media is kind of like a basal media. That means the minimal requirement for growth. That means carbon source, nitrogen source, salts, etc. Along with that, some goodies are added to this particular media. Since several goodies are added to this media, these are useful to grow nutritionally demanding bacteria or nutritionally exacting bacteria such as fastidious organisms. Now, example of such media are blood agar, chocolate agar. Blood agar is prepared by adding of 5 to 10% of sheep blood to the minimal media, and this is appropriate for the growth of Staphylococcus pneumoniae or many other organisms. Now, chocolate agar is one version of the blood agar where the plate is slightly heated and the blood cells are lysed. Among others, Haemophilus influenzae can grow in this particular media. Next, let's talk about selective media. So in selective media, we design this kind of media to suppress the growth of one category of microorganism while allowing the growth of other category. Selective media is agar-based media, and generally individual colonies can be isolated from this particular selective media. Mannitol salt agar, salt milk agar, etc. are used for recovery of specific organisms such as Staphylococcus aureus. So mannitol salt agar has been used to isolate Staphylococcus aureus, and Staphylococcus aureus colonies can ferment mannitol, and thereby they have this characteristic yellow color in these colonies. More examples involve MacConkey agar, which is selective as well as differential culture media. So it is designed to isolate gram-negative and enteric bacteria and differentiate them from differentiate based on like lactose fermentation property. So in a MacConkey agar, all the lactose fermenters would produce a pink colored colony such as Klebsiella, Enterobacter, etc. Whereas the lactose non-fermenters such as Salmonella, Shigella would produce a yellow colony. So based on color, they can be differentiated. Campylobacter agar is another agar for selection. So it's another selective media. So Campylobacter agar is dedicated for isolation of Campylobacter jejuni, which is found from the rectal or the fecal swab. It has a cocktail of antibiotics such as vancomycin, polymyxin B. All of these would allow the growth of only Campylobacter and reduce or inhibit the growth of other bacteria in that plate. Now let's talk about another differential media, and this media is known as differential indicator media or chromogenic media. So these media are designed in such a way that different bacteria can be recognized based on their colony colors. So obviously chromogenic substrates are used. Upon metabolism, bacteria produce different colors. So in simple words, different bacterial colony on the different in that same plate would have different colors, and based on that we can discriminate them. So a simple example was mannitol salt agar, because mannitol fermentation leads to a yellow colored colony, and this is very characteristic of Staphylococcus aureus. Also, you can think of the example of MacConkey agar, where lactose fermenter versus lactose non-fermenters can be discriminated based on their colony colors. Also, we can talk about Subaroides agar, which is used for isolation of fungi such as Candida albicans, and these are frequently used in the medical laboratories to isolate fungus, not bacteria. So it's another kind of selective media. Now we can classify media based on their chemical nature. Some media which are frequently used, they are solid media. Generally they are in a plate and agar is added into these media. There are other media which are liquid media which don't have agar in that, so they don't solidify. So Robertson's cook meat is one kind of like example. So where it is used for cultivation of aerobic or anaerobic microorganism. So let's say the benefit of liquid media is you have some patient sample, but the amount of the patient sample is very less. So you have a small swab. You can dip it inside a liquid culture media and then allow it to grow for a while. And then from this particular liquid culture media, you can inoculate in a plate, in a solid plate. And then it would allow the growth and isolation of specific bacteria from that plate. Now, from an industrial perspective, liquid culture systems are really important because in industry, we can grow thousands of liters of bacterial culture to get various products, such as think about the antibiotic industry. They need to grow bacteria in bulk in order to extract antibiotics from them. Also think about a particular industry which involves production of insulin or other kind of recombinant proteins. There they need to produce huge amount of biomass. From there they can extract that particular product. So from an industrial point of view, liquid culture is really important. So overall we learned the most important type of bacterial culture media used in lab. If you like this video, give it a quick thumbs up. Don't forget to like.